Good afternoon. My name is Zoe Hiroshima, and I'm a junior in Honors Independent Science Research. Today, I will be presenting my year-long research project titled Using Conservation Genetics to Develop and Test Genetic Primers for a Native Hawaiian Damselfly. The conservation of endemic Hawaiian species is one of the most important and effective ways to preserve Hawaii's vibrant culture. As a result of human intervention, native species have become endangered, which could be detrimental culturally. One way to reverse this damaging process is through conservation genetics. Conservation genetics is the analysis of genetic factors that can affect the risk of extinction in certain species. Using conservation genetics, scientists can measure and preserve the genetic diversity of populations, which is an important factor to the survivability of a species. The subject that I focused on in my research is a species of the endemic Megalagrian damselfly, specifically Megalagrian vagabundum, the scarlet Kauai damselfly. M. vagabundum is a Hawaiian species of damselfly that is native to Kauai and can be found in a variety of habitats, ranging from aquatic to terrestrial. In previous research conducted by Elizabeth Henry in 2017, she tested M. vagabundum to find its population structure, which is an element of conservation genetics that helps to determine the genetic structure of certain populations of a species and how they relate to each other. She accomplished this by using a gene called CO2. Her research concluded that the populations she sampled were very homogeneous and therefore were not at risk of extinction. In this specific figure from her paper, the bigger circle relates to the high frequency of a haplotype, with each color representing a population of M. vagabundum. One can see that the majority of haplotypes are shared within the different populations, and thus the populations are not very differentiated. Because the populations researched were fairly homogeneous when testing one gene, one might wonder how the results might differ if we used a different, more variable, and less tested gene instead. This leads me to my research question and goals for my project. After doing my preliminary research, the question I wanted to answer for my own research project was, how can I analyze a more variable gene in Megalagrian vagabundum that could result in having more genetic differentiation between populations? Initially, like Elizabeth Henry's research, I also wanted to find the population structure of M. vagabundum using the more variable gene that I would find. However, throughout the data collecting process, I had to change my goals. To accomplish my research question, I had 3 goals. Number 1, to use a previously studied and more well-known gene called elongation factor 1 to test on M. vagabundum populations. Number 2, to develop my own genetic primer for a more variable gene other than EF1 or CO2 that would effectively amplify different populations of M. vagabundum. Number 3, testing the primers I developed on samples to find variability throughout populations. Starting off with my methods for my first goal, which was to use a well-known and less variable gene called elongation factor 1 to test on the M. vagabundum samples from different populations. To accomplish this goal, one must understand the procedures I used throughout my research process. The main procedures that I conducted during my research were PCR, or polymerase chain reaction testing, and gel electrophoresis. PCR is a process used to amplify DNA samples, which can then be used to compare with other amplified samples to see the differences between them. This method that I used to PCR using elongation factor 1 stayed constant throughout my research. The samples that I obtained already had the DNA extracted from them, so I used PCR to amplify and sequence the DNA. I mixed a ratio of PCR water, forward and reverse primers, and 1 tack multiplied by how many samples I was amplifying, plus 1 control sample and 1 extra tube. After this master mix was completed, I added the DNA sample that I wanted to amplify. I amplified the DNA by using a thermocycler at specific heating and cooling conditions. This procedure was used to methodically heat and cool the primers and DNA so that the DNA would replicate, allowing me to then use gel electrophoresis. This diagram shows the PCR amplification process, where the primer, represented by the blue and red blocks, are running along the DNA strands and replicating them through the temperature changes in the thermocycler. Gel electrophoresis is a process in which electricity runs through agarose gel, shown on the right, in order to separate the DNA by size. In my methods, I ran the M. vagabundum samples for 20 to 30 minutes at 80 to 95 volts. This allowed me to see whether I amplified the correct gene, where I then sent the amplified samples to a lab for sequencing. I then received digital copies of the DNA that I could use to compare with another sample to search for differentiation. The results of using elongation factor 1 primers showed mixed results, with some of the samples amplifying and some not. The darker black lines indicate a strong amplification that I was able to send off. The more transparent or smeared lines indicate a weak amplification, and the samples with no line showed that there was no amplification. Of the samples that did amplify, there was little to no differentiation, which confirmed what I thought about there not being enough variability in this gene. In the picture shown, there are 5 rows, which represent the successfully amplified samples of M. vagabundum when analyzing elongation factor 1. These were aligned in order to locate genetic variability between them. The green bar above the alignment shows that the samples have virtually the same genetic makeup and are almost 100% identical. After doing many tests using the elongation factor 1 primers, I then moved on to my second goal, which was to develop my own primers using a more variable and relatively untested gene. My research yielded that the mitochondrial control region had potential in being a genetic primer for damselflies, as it was found to have higher variability between different populations. The control region, as shown in the picture, is at the very end of the mitochondrial genome. However, there were no existing control region primers for M. vagabundum, thus I had to develop my own. To develop the control region primer, I needed to find variable and invariable segments of the megalagrian mitochondrial genome, which is all of the DNA in the mitochondria. However, the species of megalagrian is not often tested, so there were no full genomes available for me to use. Instead, I used a species of damselfly called Euphea, which is related to the species of megalagrian. On the full genome, I found the control region, then searched for variable and conserved segments. Variable means that the DNA strand that consists of ATGC bases does not match the makeup of the strands that it is aligned with. In the results for EF1, the line above that indicated the similarities between the samples was almost all green, showing that the samples were almost identical. However, in the top picture in this slide, representing a variable region in the control region, the bar ranges from green to red, with many different colors showing the differentiation between the ATGC bases. For variable regions, I attempted to find regions of about 600 to 800 base pairs. The conserved segments look like the picture on the bottom, with a solid green line indicating no variability between the samples. For these, I looked for invariable regions of about 20 to 40 base pairs. This figure represents the structure of the primers and variable regions. The conserved segments of DNA are the darker boxes, while the light dashed box represents the variable segment that I am trying to amplify. There are 2 primers for every variable region, one running forward and one running reverse. To find the sequences for the primers, I copied the invariable or conserved regions and then pasted them into a program called Primer3, giving me 4 different possible primers for each non-variable region. Out of the 4 primers Primer3 gave me for each surrounding sequence, I chose 2, incorporating factors like GC percentage and annealing temperature into my decisions, both of which are factors that would affect the compatibility of the forward and reverse primers. All in all, this process left me with 8 total primers, 4 forward running and 4 reverse running. After developing the sequences for these 8 primers, I ordered and shipped them to the lab, where I could then use them in testing. Thus, I was able to accomplish my second goal of developing my own genetic primers for a more variable gene in the mitochondrial genome of M. vagabundum. My final goal was to test the primers I developed to make sure they amplified the correct region of the genome and to possibly find genetic variability between populations. To determine which control region primers worked best for the DNA that I acquired, I ran a sequence of trial and error tests using the same PCR and gel electrophoresis methods as before to determine A, which 2 primers were the most compatible, and B, the most effective annealing temperature, DNA amount, and time and voltage in the gel electrophoresis. These tests resulted in finding out that primers MEG148DB_F and meg1b_r were the most compatible, and the samples best visualized under 95 volts for 20 minutes. After sending off the successfully amplified samples to be sequenced, I aligned them to the original Euphea genome to determine whether the primers I developed amplified the correct region. As shown in the picture, the amplified regions do line up with the original genome, which means that the primers were successful. The purpose of this research was to develop my own genetic primers for a relatively new and untested extremely variable gene, the mitochondrial control region, as well as test for using these developed primers as well as more common and less variable primers, the nuclear EF1. I wanted to be able to research the genetic structure for this native Hawaiian species that has never been tested, for this native Hawaiian species for conservation reasons, and so that I would be able to provide research about a gene that has never been tested for this species before and does not even have primers. Regarding this purpose, I think that I was able to complete most of this to some extent. I was really successful at creating my own control region primers, as the first tests I sampled, the amplifications came out strong. As I continued to test these two primers, I achieved varied results, but the fact that they amplified in themselves is an achievement. When amplifying the M. vagabundum samples using my developed control region primers and searching for mutations, the samples tested did not yield strong or significant mutations. However, the primers that I developed did amplify the correct region, which is significant research, as there was no existing primer beforehand. Overall, the research that I accomplished could be used for further research purposes, like using the control region primers to continue searching for the population structure of M. vagabundum. Honors independent science research has been such an incredible opportunity for me to have experienced. My advice for doing this class is to thoroughly research and love your topic. You're going to be spending a lot of time doing it, so you should make sure that you genuinely enjoy every aspect of it. Also, if your research doesn't come out the way you expected it to, try not to be disappointed. Personally, I had big goals for somebody who is brand new to the research process and high expectations. Even though my initial goals weren't exactly reached, I still made a great project. Lastly, I learned so much about myself, and I discovered that I love doing research, and I was able to discover my passions for science. I want to thank my family for supporting me through this process, my friends who have constantly stayed by my side and were always willing to look over my presentations and papers, my mentor, Dr. Butler, for talking with me and providing my research samples, and Dr. Chan for answering every question I had and teaching me throughout the entirety of my research. Also, thank you to everyone who came to watch my presentation. Thank you all so much. And here's my references. Thank you.